Hi everybody, welcome to another Chats with Chaudhry, and today I'm delighted to be joined by my good friend, officially known as Nando, Ferran Sanchez, but Ferran to me, who is my fellow vlogger, but more importantly is the Market Development Manager for Pharma CRO, brackets MS and CE, and Aftermarkets EMAI at SCIEX. What a long job title. I'd hate to print your business card by hand, Ferran. Anyway, exactly. So, Ferran, it's lovely to see you. And today we're going to be talking about a topic which was in every life science publication, every webinar before COVID hit in March. This was the #1 topic I think people were talking about. And it was got a bit quiet because everyone's been focusing on vaccines. But we're going to be talking about today, which is even if I can say it properly, nitrosamines and nitrosamine analysis. So, Ferran, first of all, hola, my friend. How are you and how is life in Spain?
I'm good, thank you. Life in Catalonia is quite good. So, I mean, considering COVID, so we are almost starting a kind of opening after being for a few months. I will not say totally closed, but yes, almost closed. So then I do believe that they are starting to open thanks to the vaccines in first, and also because the cases are just going down, and especially the hospitals are not under pressure as they want. So, but yeah, COVID, as you mentioned before, COVID now is our main concern, not only because of the science, but more for our day-to-day. But yeah, it's going well. Thank you. Yeah.
Good. Okay. Well, it's getting better here in the UK as well. I think we've had well over 20 million people vaccinated, so it's all good. And actually, I noticed, people are wondering in Ferran's background on just to his left of his shoulder, those are actually framed album covers, so which are pretty cool. So we might do a competition at the end and see if you can guess any of them and send them in as part of the conversation at the end. So
we can do it.
So Ferran, before we talk about nitrosamines and their importance, for those who are not familiar with SCIEX, could you just give a very quick overview of SCIEX and also your background as well?
Yeah, SCIEX is a company that we are offering hardware, software, and complete solutions for any kind of market. So I would say related to mass spectrometry. So in my case, I'm responsible for the pharma market. So that means, as you mentioned before, pharma and CRO. So, and we are offering solutions from the research and discovery until the QC, QA, and manufacturing, previous to just release the drugs into the market. Then, yeah, SCIEX is heavily involved into the pharma industry. We have been leading this market for many, many years, especially what concerns to quantification. And yeah, nitrosamines is one of the cases in where so then we are just trying to just well, yeah, not trying. We are delivering, in fact, solutions for the customers. I will not say only instruments because it's very important to deliver the right solution and the right answer to the customer in that case, for especially for giving the answers to the authorities when they are requesting or they have any auditory in that case. Yeah.
Brilliant, thank you. So let's talk about nitrosamines. First of all, being a non-scientist, what is a nitrosamine and why are they so important?
Well, nitrosamine, basically, they are important. Well, nitrosamine is an impurity, in fact. So, and that's why it's important, because every company needs to report the impurities in their APIs and in their final product, so then the formulated products. And in that case, the importance of the nitrosamine is because it's considered as a genotoxic. So that means it can cause cancer at the end. So it's important not only to, I would say, to identify and quantify your nitrosamines in your compounds, but also to really know the daily intake or the concentration that it's coming into the body. That's why it's important. Yeah.
Right. Okay. And what are the main challenges and difficulties then when it comes to doing can't even get it right, nitrosamine analysis? What are the main difficult challenges when you're actually having to analyze them?
Yeah. The nitrosamines, they have different precedences when you are manufacturing a pharmaceutical product. So then first is to do the risk assessment, and this is something that the authorities, so that means the MEA and FDA, they are requesting to all producers of API, but also formulated drugs, to have a risk assessment about where in their process, manufacturing process, they will find or they can produce nitrosamines, right? Because this is coming from the manufacturing process.
Right.
So
Okay.
this is one of the difficulties, just to know into your process where you have to pick the samples and analyze the samples at the right time to avoid to have nitrosamines during the process. This is one. Then in what concerns to the analysis of the nitrosamines, so it's because of the I would say the presence in the API. So then how difficult is to do the sample preparation? So because the API can just hide the signal of the nitrosamine in some way.
Right. Okay.
Right? So then it's sample prep and then the chromatography. That's why it's important to have in your team people that it's ready just to offer solutions in that case, right? So then I will say it's not only what you can find on the MS, what is important on mass spectrometry is also what are you introducing. If you are introducing nothing into the mass spec, you will not have a signal. So then everything is related. That's why, as an example, we are working very close with Phenomenex, our colleagues from Phenomenex, developing good sample preparation methods, but also to choose the right column to separate all the nitrosamines in the right manner at the right time. Okay.
Right. Now, this is a big question because you were obviously talking about the fact that you can find them at different stages of the sort of development process.
Yeah.
So obviously there will be different challenges and techniques you're going to need to use at those different stages. So what are the most suitable technologies for overcoming these challenges, and why are they the most suitable?
Yeah, there is different techniques that you can use. I mean, so usually, so in the past, people were using UV or LCUV, but also GCMS. But now, in the last, I would say, 5 years, LCMS. So then it's probably the technique more used for the identification and the quantification of the nitrosamines because of the sensitivity, specificity, and robustness. So usually what people is using the most is triple quad technology, again, because of the power of quantification of those instruments.
Right.
And especially when you are looking into or when you are moving from the method development into the QC, where you have just to run samples and samples and samples then. So it's a very easy instrument to use. It's very robust, and it's giving to you the sensitivity that you need, and probably more, for the quantification of the nitrosamines. It is true that for some specific cases, people it's using or we can recommend also different technologies, right? So then I would say if you are more on the method development, probably you would like to have a complementary or some complementary technologies, and you can have Q-track technology as an example, but also high resolution, yeah. And some of the authorities, they are requesting for having high resolution as one of the instruments to use, but also something that is complementary to triple quads in our case, that it's ion mobility for having a better separation and a better, I would say, signal, a clean signal at the end of the process when you are looking after very, very, very low concentrations. So say that, and as a summary, so if I have to recommend an instrument to a customer, will be more a triple quad at the first instance. And then it's depending about in which stage of the, let's say, development of their drug they are. So then if they are more on development and they want something more complementary or something that it's more, how to tell this, that can do more things, then we go to Q-trap or to Q-TOF technology. Having ion mobility, it's very depending. Yeah.
Right. Okay. And what other areas can this technology also help with? Because not just the nitrosamine analysis.
Well, it can be used in, I mean, so if we think in bioanalysis as an example, so it's the most instrument or the instrument more used. So then, I mean, triple quads for bioanalysis in the pharma industry, it's the instrument of choice. So then, but in any area when you were talking about the development of a drug, so you can use mass spectrometry. So for the discovery, for the characterization, and for the quantification. So the thing that when we are talking about R&D, so as an example, so R&D, when they are starting to identify the different targets, you need to use mass spectrometry for doing metabolomics, lipidomics, proteomics studies to complement and to really accelerate the way in where you are identifying targets and the target of the drug, right? So then when we are looking into the development, so you need to develop methods again just to quantify the drug itself, but also is when you start to do preclinical studies, as an example. Then you need to identify all the possible metabolites, toxics, and it's here when, as an example, we are talking about nitrosamines. You need to know the kinetics of the drug. So then, and all those studies are developed and done on mass spec. And then when you go into the clinical studies, it's mass spec again. Right. So, and then we move into the production at the end. So then, and it's quality controlling where mass spec is also used as well. So then that's why it's a wide, or it's a technique that is widely used in all the process. Right.
Right. Okay. And looking ahead now for the future, what future trends can you see around nitrosamine analysis?
Yeah, around nitrosamine analysis. So then, so just to give a bit of history. So then they started with few cases in June, July 2018, and it was, people was thinking it was few cases, and then it was something like a bomb, because it was affecting all the drugs that they call sartans. So sartan, valsartan, ivarsartan, all the sartan drugs. So then, and then it's jumping into more drugs. So then at the end, what the authorities are requesting is almost a kind of evaluation of the risk assessment in any drug that can have a nitro group, right? So then that means that, yeah, to me, the future on the nitrosamines is that they will continue pushing for new drugs and to analyze and reanalyze every single compound. In addition, so when you look about the advisors, that doesn't mean that it's regulated, but the authorities that are advising inside of this risk assessment just to do analysis, as an example of the water. So analyze the water that you are using in production, just in case you have, nitrosamines on this water that then at the end, so then can affect in your final product.
Right. Okay.
Even other products that you must think that probably they don't have, or nitrosamine is the biologics compound. So we are thinking of a biologic compound that is created in cells. You might think, so, well, should not have nitrosamines. Authorities are recommending this, and probably they are more recommending for what it is, synthetic biology. That means more probably related to synthetic peptides and things like. So then, yeah. So in the future, this is one. So then the panel of drugs will be expanded, and this will continue being a hot topic and a case. And then analytically, I will say that what is important is just, and because one of your first questions, so it's, what is the difficulty? The difficulty is that you have different type of drugs, different type of APIs and final products, and it's very difficult to establish something like a universal method. And it is where, as an example, we have just released a technote recently in where we are analyzing 6 drugs, 9 nitrosamine on the same method. And this is key, because in that way, so then you need to do less development for every single drug. So you have a universal method that you can apply for all the drugs, all the process is more easy. So, and at the end, so if the process is easy, that means time and ultimately money for the company. That's key. Yeah, for the pharma industry. Yeah.
Yeah, sure. That makes sense. Well, thank you for doing that, actually, because as I said, I've saw so many webinars and papers and articles on this. But as I said, then COVID hit, and this suddenly took a back burner, because understandably, because people were focusing on vaccines and diagnostics and so on. But as you said, and I didn't understand actually why it become such an issue, and I was seeing so much of it, like you said, late 2018, 2019, I started seeing a lot more in terms of the media and on the websites and so on, and on LinkedIn about this as a topic. So thank you for sharing that. So if people want to know more about the analysis techniques that you were talking about, where can they get more information?
Yeah, I will share with you some links. So because one link is a kind of, well, we have a landing page where people can find different technotes related to nitrosamines, not only from SCIEX, but also related to phenomenics as well. So that means columns and so on. A very interesting technote that we have just released, and it's this one, that it's, I would say, is boasting in some way what the Pharmacopoeia is telling to us. So then it's really improving the Pharmacopoeia method with these 6 drugs in one method, 9 nitrosamines, that it's with very nice LOQs and LODs. and I will just send to you the link as well.
Fine.
And finally, and it's depending on the region. So then, because now starting on the 22nd of March, so that means in 10 days practically. So we are organizing a series of webinars. So
Right, okay.
22nd, 23rd, 24, and 25. So in local languages. So then we want to be more close to our customers. That's why we are organizing first in Spain, second in France, second in Germany, sorry, third in France, and fourth in Italy. And in that order, as Bale will tell.
I was laughing. I was wondering you were going to say
Yeah.
that. Yeah.
Gareth Bale will tell in that order. So we are organizing those webinars in where we will have very interesting conversations from Phenomenex, from SCIEX, but also from customers talking about their experience on the analysis of nitrosamines and on the risk assessment on the nitrosamines. That it's one of the most important parts before to run the samples. Because again, so first you need to know where to take the samples to have the right answer in some cases. Yeah.
Fantastic. All right. Well, look, those links, viewers, will be above the video, so you don't have. That's why Ferran's not read them out, because they take too long to read them anyway. But we'll put them above the video with all the links, so you can click to go to the landing page, look at the tech notes, and also obviously register for the up and coming webinars in your local language, which is great. So there you go. They're trying to think of everything, so you can check those out. And Ferran, just grab one album cover from there and just put it up, and we're going to ask people to try and not give a guess right now, obviously, but they can actually put a comment below the video. Just pick one cover, and then we're going to ask people to try and guess.
I will choose one that is easy. Okay. I will choose another one, but I do believe so. Yeah. Okay, let's choose this one. The song is very well recognized. Okay.
All right. So just put it up to the. All right. Okay. So if you know the name of that album and the band, put it in the comments below the video, as well as, of course, more importantly, leaving any questions for Ferran about nitrosamine analysis. And actually, one final note, Ferran, as I've got you, and I very rarely get to speak to you because you're always such a busy man, what's your predictions for the NBA finals this year, then, as the season's kicked off?
Good question.
Brooklyn must be the favourites now. They got hated. We won't talk about it.
Brooklyn is on the list. So, but I will say for the finals this year, I see Lakers again, unfortunately for me, but fortunately for you. But Lakers will reach the finals this year because some changes are coming in the Lakers for the good. So I was reading some names that they can go to the Lakers. And yeah, we'll be happy that Marc Gasol will win his second ring, even if it's with the Lakers.
Right. And who will they be playing?
Yeah. And in that case, Milwaukee.
Well, there you go, viewers. So make sure you remember this from Ferran. He said Lakers versus the Bucks in the NBA finals this year, with Lakers to win it. So Ferran, as always, it's a joy to see you, and it's lovely to speak to you. Thank you very much. Genuinely, I've learned a lot today. Hopefully everyone else has as well. I can get more information, as I said, from the links above. If you know what that album cover is, put it in the comments below as well. If you've got questions for Ferran about nitrosamines, put it on the comments below. And if you disagree with Ferran's selection about the NBA finals, put it in the comments below. So on that note, Ferran, I wish you a fantastic 2021. I hope you and your family and everyone at SCIEX have a fantastic year. And more importantly, you're all safe and happy and healthy. It's great to catch up with you again, my friend. Look after
Thank you. Thank you very much. I wish you the best as well.
And also check out his vlog, because he's got a very good vlog as well, where he talks to lots of scientists. So check Ferran's vlog out as well. And everyone else, I hope you enjoyed that. Until next time, as always, stay well and stay safe. Bye-bye.
Thank you. Bye.